Hi everyone, so in this video I'll be explaining translation in a very simplified manner. So basically translation consists of three main stages. So first one is the initiation stage, second one is the elongation, and then thirdly we have the termination phase. So first we'll look at the initiation process. So for initiation, what happens first is that we have the small ribosomal subunit, which moves, that is when a protein is about to be expressed. The small ribosomal subunit moves through the mRNA, and then it binds to the place where we have the start codon. As you can see in this diagram here, here's where we have the start codon, and then the small ribosomal subunit will bind to it. And then once the ribosomal subunit the small ribosomal subunit has been bound to it, next it is the tRNA, which has the anticodon, or the codons that are complementary, like what you have here, that are complementary to the start codon. So we say then, and of course that one it carries the methionine amino acid. So, and of course the process requires energy. So lastly, it's the large ribosomal subunit that will bind to it. So the large ribosomal subunit contains three main elements which we need to note. It contains the A site, the P site, and the E site. So all these three sites are very crucial when you'll be learning the translation process, especially in the elongation part. So the methionine will be bound at the P site. So that is very important to note. So I'll repeat again. In the large ribosomal subunit, we have the A site, the P site, and the E site. So the A site is where the amino acid comes initially, and then of course it now moves to the P site, and then the E site is where it exits. But in this case, in the initiation stage, the methionine, when the large ribosomal subunit binds, the tRNA carrying methionine will automatically will be at the P site, that is, to create room for the next amino acid that will bind at the A site. So next it's the elongation phase. So in the elongation phase now, here is where now we'll have the amino acids that we are talking about that will now bind to the A site that we had looked at initially. Remember we had said that in the large ribosomal subunit we have the A site, the P site, and then we have the E site. So the next amino acid that is that has an anticodon that is complementary to this codon on the mRNA will be bound at the A site. And of course here, remember, we'll have there's methionine, but of course now there are other amino acids that have formed peptide chains and everything. So the important thing is just to understand what happens at the A site, at the P site, and at the E site. So firstly, an amino acid that has a site that is an anticodon that is complementary to that of the mRNA will bind to the A site, and then after that we'll have the a peptide bond that will be formed, and of course this peptide bond, the formation of this peptide bond will be catalyzed by the rRNA. Remember we have three types of RNAs. So the rRNAs in the large ribosomal subunit will catalyze the formation of the peptide bond between the amino acid in the A site and the amino acid in the P site. So the bond will be formed here. So next, once the amino acid, the bond has been formed, so the next in the next part, then this bond here will be broken, or the amino acid, the tRNA at the A side will be detached from the amino acid, and then finally the amino acids will move to the tRNA will move to the E site, and then the tRNA carrying the polypeptide chain will move to the P site, and of course now the process will continue. Again, another amino acid will come to the A site, and then of course now it will interact with the amino acid at the P site, and the rRNA will catalyze the formation of a peptide bond, and once the peptide bond is formed, then of course now the amino acid polypeptide chain will be detached, or the tRNA at the P site will be detached from the amino acid chain, and then of course now as the processes require energy, and of course now the tRNA will be released, and so forth. So we can also look at it in terms of 2D, which would make it easier to understand. So when we look at it, when we look at the 2D illustration, here we have the first amino acid, which is methionine, and it has an anticodon. Now this is what we call an anticodon, that is complementary to the codon, to the start codon on the mRNA. Now this is our mRNA, so of course now it will bind here, and then next we'll have the second amino acid, which will now bind to the other codon now, which is complementary to though the selection will be on the basis of its complementarity. Now because this one has an anticodon that is complementary to this codon here, it will bind. And then next we'll now have methionine here and alanine here, so a peptide bond will be formed between them. So of course once a peptide bond is formed, the amide at the A site, at the P site, if you can remember from the previous diagram, will now be detached, and then the one here that is the one that is at the A site will now move to the P site, and the one at the P site will move to the E site, and then it will be released. So we can see that one here. So we've seen this one will move at the E site, and of course it will be released. This one will now move to the P site, and of course now it will now be carrying the amino acid chain, and of course now a new amino acid that is complementary to the codon that is exposed at the A site and on the mRNA will now bind, and of course now the process will continue as you said initially. And of course now that's why we call it the elongation phase, because as the process continues, the amino acid chain will continue to increase in length until the termination phase. So lastly we have the termination phase. Now this is where the process of translation is terminated. So when the ribosomes reach the stop codons and the one on the mRNA are the ones that mark the end of translation process. So at the stop codon, one thing we need to note is that there is no corresponding amino acid. Remember, for these other codons, we have amino acids that the amino acids that are usually attached to tRNA, which have anticodons that correspond to the which have anticodons that corresponds to the codon on the mRNA. So for the stop codon, we don't have amino acids that correspond to the codons. So in that case, the release factor will be bound at the A site instead. And once the release factor is bound, the complex will dissociate, and the process, of course, of translation will be terminated. So thank you very much for listening to this video. Kindly subscribe, share with your friends, and don't hesitate to ask any question on the comment section. And if you need any form of tutoring, you can contact me through the email displayed. Okay, have a good day.